Okay, so this is the third installment of the Evolution of Populations lecture. Try to make this all fit into one. The next topic of discussion is the interactions among evolutionary forces. So here the idea that we need to think about is that natural selection might actually be in competition with mutation, although it's not at a high enough rate to really counter its effects. Genetic drift can also act to remove genetic variation from a population. Genetic drift should only outweigh natural selection in a small population, though, not in a large one. A study was done looking at the copper resistance allele in a certain grass that grows in Great Britain. And this is showing us how natural selection and gene flow can kind of be in competition. Move this a little bit so we can see it better. What was discovered is that some grass plants that grow in an area where there was a copper mine actually possess an allele that makes them tolerant to copper. However, this allele also stunts their growth if they are in normal or unpolluted soil. So it makes sense that we would see evidence of selection for copper tolerance on mining sites and selection against copper tolerance off mining sites. Thus, we would expect to see 100% copper tolerance allele in mining sites and 0% in non-mining sites. But that's not what they found. Quite interestingly, a little bit kind of upwind and a lot downwind from the site, they actually do see the presence of that copper tolerance allele. And really what it just gets down to is pollen being carried by the wind. So we do see plants off the mining sites containing this tolerance allele, even though it affects their growth. So here, gene flow is going to be not really overriding natural selection, but competing with it. And this is just measuring the frequency of those alleles in the population. So the message here is that allele frequencies sometimes reflect a balance between opposing processes, such as gene flow and natural selection. In such cases, observed frequencies will depend on the relative strength of the process. So for example, if this prevailing wind got stronger for a long period of time, we would probably see that allele much farther out than we already do. Now, natural selection can also act to maintain variation. So let's take a look at that. In other words, keep the variation where it is. Here we're going to look at frequency-dependent selection first. Frequency-dependent selection is the fitness of a phenotype will depend on the frequency in a population. So in other words, whether you are common or not. There are two types. There's negative frequency-dependent selection. I guess that was fine. And positive frequency-dependent selection. So with negative frequency-dependent selection, the rare forms have a selective advantage. Therefore, their alleles are kept in the population and the variation is maintained. An example right here is about water boatmen. We've all seen these. They occur in 3 colors. And the rare forms are less preyed upon as predators have a certain search image in their brain about what their prey look like. And this graph represents a study that was done where water boatmen were put in various proportions of color in tanks with predators, and every time the most common water boatman was the most heavily preyed upon. And you know, maybe you would think about, well, of course there's more of them, but those that were rarer experienced a lower frequency of predation because of the search image that was formed by the predators. A second example of negative frequency dependent selection is having different resource requirements that may maintain variation as well. Plants with different resource requirements than the more common forms will actually be selected for because they don't experience as much competition. The next type of frequency-dependent selection is positive frequency-dependent selection. This is that the common forms are selected for and eliminating rare forms within the population. So in this case, this is just an example with a little picture here where the more common form of butterfly or moth is actually selected for as the rarer one, which stands out as the one that's picked off by the predator. The next pattern of natural selection is called oscillating selection. Now, oscillating selection is basically exactly what it sounds like. where we have an alternating pattern of phenotype selection, and this is going to work to maintain genetic variation in a population. And environmental changes can cause these swings in selection. And a really cool example is about some ground finches, the medium ground finch on the Galapagos Islands. Follow along this graph here as I talk about the text. What we look at is beak depth. In the drought years, notice that the beak depth increases because there aren't very many small soft seeds. In fact, the seeds are larger and harder, so the large bills are favored. In wet years, like here, there is an abundance of small soft seeds. Therefore, having a small bill is actually favored, and you have more precision in getting at the nutrients inside of those seeds with a smaller beak. Another pattern that we see is something called heterozygote advantage. And I talked a bit about this with you guys when we covered some of our crosses. Now, the heterozygote advantage is where the favoring of heterozygotes occurs over homozygotes. And this works to maintain both alleles in the population. Okay, so if it's advantageous to be a heterozygote, it would make sense that we are going to keep both of the alleles in the population. Now, sickle cell anemia is given the allele S. There's a mutation in that allele that causes red blood cells to sickle, which we've discussed, and that causes severe anemia because those blood cells can't carry oxygen as well as normal blood cells. The S allele has a frequency of 0.12 in Central African populations. In fact, 1 in 5 individuals is heterozygous. 1 in 100 is homozygous. And it is a fatal form of the disorder. They almost never reproduce because they die before their reproductive age. But heterozygotes are less susceptible to malaria. So you might ask, well, why then wouldn't the homozygotes, since they die, why wouldn't we just lose that allele altogether and eventually, not have this problem? Well, the problem is that if you are a heterozygote, you are actually more resistant to the parasite that causes malaria. Now let me talk about malaria for a second. Malaria is caused by a plasmodium. It's a parasite, and it causes also the red blood cells to sickle. But these cells can be filtered out by the spleen. However, if you are a homozygote for sickle cell anemia, you have too many red blood cells that are trying to be filtered out for your spleen because you're already sickled, and so you can't handle it. Therefore, being heterozygous for the S allele is still adaptive in areas with malaria, but not in areas without malaria. So what you see, this is very interesting. This graph shows you the incidence of sickle cell anemia in Africa, and this shows you the incidence of malaria in Africa. So notice how these two unrelated, things go together, where if you have this disorder or if you're a carrier of this disorder, you're resistant to this disease. What's also interesting is African Americans have lived for about 15 or so generations in areas very low in malaria, like in the States and other countries. Thus, we shouldn't see this S allele in such high proportions anymore. And in fact, we don't. Now, only about 1 in 375 African Americans develop sickle cell anemia, as opposed to their ancestral counterparts having a much higher rate of that. So we see that selection can maintain variation within a population in several different ways. So let's take a look at the forms of selection. And there's again the graph that you just saw. There are 3 forms of selection that this will be familiar from biology. And it's really patterns or trends of selection. First, remember that genes have multiple effects. This is called pleiotropy, and that many traits are controlled by several genes, in other words, polygenic. Selection will operate on all genes, especially those that make the strongest contributions toward phenotypes. The first, disruptive selection. I'm going to go ahead and show this picture here, which contrasts the 3 patterns. And the way that these work is the top graph is your initial population, the red is what's being selected against, and then in the end is your resulting populations. So we'll start right over here. Disruptive selection. This is a type of selection that tends to eliminate intermediate phenotypes. as evidenced by the example of the African fire-bellied seed-cracker finch. That's a full mouth. And that bird, that's a picture of the bird there. Now these birds feed on seeds, large seeds or small seeds. The large-beaked birds open up tough shells of large seeds, and the small birds, or the small-beaked birds, can handle small seeds. But the intermediate beaked birds are really kind of out of the loop because there aren't seeds in their habitat of an intermediate size. So over time, what you see is two peaks forming in terms of which body size or which beak size is going to work best in this habitat. And so over time, you might expect these two to become so dissimilar that they could no longer interbreed if they keep to themselves. The next type of selection is directional selection. Directional selection is a kind of selection that tends to eliminate one extreme from the group of phenotypes. So it will actually be reducing the alleles that create the phenotype. There was an experiment involving Drosophila, our fun fruit flies, in which researchers removed flies in each generation that would move toward light. So they were just pointing out a behavior and taking out the flies that exhibited that behavior. They then using only the remaining flies as the parents. So over time, the average number of flies that would move toward the light decreased with each generation. So with directional selection, what you're looking at is you're selecting against one extreme or the other, and then shifting the entire population one direction or the other. Next is stabilizing selection. And it does kind of what it sounds like. Stabilizing selection is a kind of selection that tends to eliminate both extremes from a group of phenotypes. It prevents deviation from the common intermediate phenotype. A very interesting example is human infant birth size and how it's correlated with survival rate. Infants that are born too small or too large have lower survival rates, and especially in areas of our world that have no or little medical care. They survive less often than do infants born of average birth weight, and that's a graph shown here. We actually see the same pattern for hatching success in duck and chicken eggs, so we can't really breed our chickens to have more eggs because it makes, well, we'll find out later why, but it makes them have lower reproductive success as it turns out. So stabilizing selection looks like this, where you tend to select against the extremes and narrow the peak instead. Make sure you know the difference between these three types of selection. And next, what we're going to look at is a specific example of selection. This is in the color of guppies. Guppies, how exciting, right? Well, typically evolutionary biologists will study what's happened in the past. We rely on observation versus experimentation. Darwin, like most traditional evolutionary biologists, assumed that evolution occurs at a very slow pace. However, that's not always true. This allows experimental studies to be carried out, and some of these can be field-based, which is, nature. So let's take a look at guppies as our example. Guppies live in different environments. They live in streams in northeastern South America and Trinidad, and are capable of colonizing the streams above waterfalls. They can rely on floods and other secondary channels to move upstream above waterfalls. Now they have two potential predators. There's the killifish, this guy here, which can actually wriggle and swim its way upstream, even in just the rain, and be in these upper pools with them, but they just occasionally feed on guppies. Now the pike cichlid lives in the lower pools, cannot colonize the streams above the waterfalls. And pike are definitely piscivorous, they eat fish, but they remain in the lower pools. But guppies live in both pools above and below the waterfalls. The guppies that live below the waterfalls are preyed upon by pike, and that's a very high predation rate. But those guppies that live in the pools above the waterfalls that can be subject to predation by the killifish, that rate is actually pretty low. So let's take a look at an experiment that was conducted to study this phenomenon. The high predation pool guppies, remember that's below the waterfall, it was observed that they have very drab coloration. They reproduce at a younger age and the adults don't get very large. There were different selection pressures than in the pools above the waterfalls. Up there, natural selection would, down low, the natural selection would act against bright coloration and late maturation. The low predation pool guppies above the waterfalls had bright coloration in the males, later maturation, and larger adult size. Up there, the males were better at being territorial and at mating. The females were larger and laid more eggs. And natural selection favored individuals who could better compete for mates and generate more offspring. Natural selection favors individuals who can better compete for mates and generate more offspring. So these were just the observations out in the wild. So what about the lab experiment? Endler, now at UC Santa Barbara, placed guppies in pools, about 200 per pool. He had a total of 10 pools. Then 6 months later, placed pike cichlids in 4 of the pools, killifish to 4 other pools, and left 2 pools as controls with no predation. 14 months later, 10 generations, he compared the populations. The guppies that were in the no predation pools, which are the controls, and the low predation pools, the killifish, were indistinguishable, could not tell them apart. They both were containing large fish with bright coloration. Remember, these all came from the same group of guppies. The guppies in the high predation pools with the pike ended up smaller and drab in coloration. Now, guppies don't live very long, so these aren't the exact same fish. These are their offspring, their offspring's offspring, etc. Now, this showed that predation led to rapid evolutionary change. But is this what actually occurs in nature? Remember, it was just done in the lab. So what about out in the field? Sorry, that just shows what we were looking at prior. Okay, there's the picture of these pools. So out in the field, Endler and Resnick at UC Riverside observed guppy populations below the waterfalls, but there were none above. So they found a natural environment where there were guppy below waterfalls, but not above the waterfalls. Now, they did find pike in the lower pools and killifish in above waterfalls. So they basically had the same situation that they created in the lab. And what Resnick and his team with Endler did is they transplanted some of the guppies to the pools above the waterfalls. And then they would come back every 7 years for observations. So they found a natural setting with these drab-colored guppies in the lower pools, and then they put some of them in the upper pools where there were killifish. Upon their observations, they discovered that the upper pool guppy populations, that is funny to say, rapidly evolved with characteristics of lab-based low predation guppies. So what happened is these fish had later maturation, a bigger body size, and brighter colors. And the differences, when they analyzed them, were genetically based. And the evolutionary change occurred in less than 12 years. Very, very interesting, the fact that they saw that occur so quickly. Now, evolutionary biology is a historical science. Nonetheless, in some circumstances, experiments can be conducted in nature to test the hypotheses about how evolution occurs. Such studies often reveal that natural selection can, in fact, lead to rapid evolutionary change. I'm going to stop this here and do one last short installment.